All right, some tough questions there. We'll move it right along. Maybe we'll get some more answers with the next speaker. Brian Lee from the Allen Institute will be speaking about multimodal properties of developing human cortical neurons. Brian is co-leading the PatchSeq pipeline at the Allen Institute and has really taken the lead to push the pipeline into the direction of the juvenile human surgical collections and feeding into the PatchSeq pipeline. So welcome, Brian.
All right, thank you, Jonathan, and thank you to the organizers for setting up this event. I'm pretty excited to share some of the stuff that we're working on. It's nice to get the introduction, so I don't have to do the introduction from C.E.T.S about some of the developmental properties, but I'm going to dive a little bit into the same kind of work that both him and I are aligned on. Okay, so just to give a brief introduction about some of the human work that we've done. Over the course of, since this project has started in 2017, we've had a collection of papers, let's see which one is, a collection of papers examining the functional properties of neurons in the human neocortex, and these papers are divided up into specific types of cortical neurons. So Tom Chartrand and Brian Kalmbach led a paper looking at layer 1 interneurons. Rachel and I have described the functional properties of interneurons in the neocortex. And then you heard a little bit about the Berg paper that looked at layer 2-3 excitatory neurons. And then Rachel and I are working on a manuscript right now that takes a more comprehensive look at all the excitatory neurons. So what I want to highlight with this is that, with all these sampling of these neurons, it's been over 3,000 human neocortical neurons. And when we look at the Hodge taxonomy and how we identify the transcriptomic identity of neurons, over all these papers, we've been able to comprehensively characterize all the neurons in the human MTG taxonomy. So this is a really nice set of work that nice set of data that we're continuing to work on. But more importantly, this is the framework for what we are working on with the human developmental and pediatric work. So this started off as a collaboration with Seattle Children's Hospital that Ed and Jonathan helped initiate. And let's see. And so with this work, we're able to work with the neurosurgeons, and during the resection of a pathological tissue, they're able to use intraoperative navigation to identify the site of the pathology. But where it's important for us is they're also able to identify where the resected tissue that they hand off to us. And this is really important because then we can take those coordinates and the MRIs and align them to a CCF and get a very precise location of the resected tissue that we work with. So since this has started back in late 2023, we've had 49 cases, and here is the locations of all those resected tissues across the brain. And then here's the distribution of the 49 cases that we've had, ranging as young as 6 weeks old all the way up to 18 years old. And so with all these experiments, we've done PatchSeq. I'm not going to go into too much detail. It's been brought up a couple times, but essentially PatchSeq allows us to obtain the electrophysiology, morphology, and transcriptomic identity from a single neuron. And so with these 49 cases, we've been able to patch onto 1,400 neurons, and then here is the distribution of the age range from each of those neurons that we have collected. And so this is still in the works right now, but we are working on, when we get the transcriptomic identity of the neuron, we have all these different taxonomies and trying to determine what the identity of the neuron is. And so part of this work, we are using 3 different taxonomies to identify what the identity is. The human pre- and postnatal from the Arnold Kriegstein lab, our standard reference that I talked about early in the first slide, the Hodge MTG taxonomy, and then Ed gave a brief introduction about the human CAD. So these 3 taxonomies are what we're aligning our neurons to and seeing which one makes the most sense. I will say that from our collaborators and friends in South Africa, the Dorit Hockman lab, she had a nice paper that looked at the Hodge MTG taxonomy and was able to identify that a lot of the key marker genes that separate and identify the different transcriptomic types are preserved in pediatric patients and adult patients. So just real quickly, for the human pediatric work, we do patch-seq, and we do that in acute slices and culture slices. So the culture slices provide us the ability to patch on to or preserve the tissue for 2 days up to 14, 21 days, and so on. So another benefit of that is we are able to apply enhancer AAVs to those slices during that culture period and allow us to target cells in a very cell type specific manner. So the first thing that we looked at is the firing rate, and the next two slides may look very similar to what Dias showed, but the first one that we wanted to look at is the firing rate of a neuron and how that changes from a pediatric patient to an adult patient. So what I'm showing here is the voltage response or the action potential firing rate after we introduce a current to the particular neuron. And this current that we applied is standardized to the firing rate of the neurons, so that way we ensure that they are of appropriate and comparable current for each neuron. And we have divided this up into the different transcriptomic identities of the neurons. And so the top one, I'm showing layer 2-3 excitatory or IT types, and then parvalbumin, and then SST. And hopefully what you can appreciate right away is that the red traces are in the infant and the gray are from adult. You can see that there's an appreciable difference in terms of the number of action potentials that are fired in the more in the adult than are the infant. And then we can look at the FI slope, which is the slope of the firing rate, and we can plot that on a FI by age plot. And on the x-axis is age, but that is in log scale. So really highlighting the earlier time points. And what you can see is that there's an increase in the FI slope as you get towards adulthood. So this developmental trajectory kind of matches kind of what Dias showed in his previous presentation, or in his presentation. And then we can look at those slopes in the different transcriptomic types. So the layer 2-3, we have a much steeper slope, which we've categorized for this Spearman correlation as 0.49. And then the interneuron and SST are not quite as dramatic as the layer 2-3 IT. So with those firing rates, we wanted to look at a single action potential and see how those are changing over the course of development. So what I have here are the average action potential traces and then the AP phase plot for the transcriptomic types, layer 2-3 IT, p-valve, and SST. And we have the adult in gray, blue is adolescence, and the red is an infant. And hopefully what you can appreciate right away is that the AP action potential is much wider in the excitatory type compared to, or sorry, in the infant compared to the adults. And that trend is similar for all types, but then much more dramatic in the layer 2-3 IT types. And on the middle, you have the box plots just quantifying that information. And then again, we can plot the trajectory of the AP width by age, and you can see that the APs start off quite wide and then gradually becomes more narrow as you get towards adulthood. And this trend is similar for all three transcriptomic types, but then again, with the layer 2-3 IT type, it is more significant. So since this is patch-seq and we have a decent amount of data, we can start to break this down and see for particular transcriptomic identities, do they have different trajectories? So here is everything summarized in glutamatergic and GABAergic, and you can see that they both follow a similar trajectory, and then we can break these down into the different subclasses. Now I will state that, as we continue to dive deeper into this, the size of our data set diminishes a little bit, so this is just kind of a preview into what we can see. I don't feel comfortable stating that any of this is super conclusive, but this is some of the trends that we're beginning to see. And so for excitatory types, that they are kind of aligned with this developmental trajectory, and then the GABAergic types seem to be much more broad. So with chandelier cells being on this bottom one and then VIP. So I think we'll continue to collect more data to see how these different subclasses diverge in these developmental trajectories. Okay, so with the layer 2-3 IT type being the one that shows the biggest difference, we wanted to take a little bit deeper dive into can we find a functional or molecular basis for this change, given since this is patch-seq data and we have some of the genes. So this is just repeating what I showed on the previous slide. So we looked at some differential gene analysis, and what we're plotting right here is looking at infant versus adult. And you can see that with these volcano plots, there's a huge number of genes that are upregulated in the adults. But since we're looking at specifically action potential, we can look at the sodium channel genes or potassium channel genes. And what we see is we see quite a few sodium channel genes that are upregulated in the adults. We can look at adolescents versus adult, and we don't really see as many genes, and only one potassium channel gene is. So this is giving us an idea that potentially this upregulation of potassium channel genes is explaining the narrowing of the action potential. So what I'm highlighting here is this gene right here is a KNC2, which is a KV3.2, and then we can look at that individual gene and see how that is plotted either in a box plot and categorizing into the 3 different stages, or we can look at the gene by age relationship. And what you can see right away is that the KCNC2 is quite low early on and then gradually increases. Okay, so take a little step back, and so for a subset of our experiments, we've actually done a different type of analysis and experiment. So just to remind you that patch-seq, we extract the nucleus to get the transcriptomic content, but for a subset of these experiments, we're actually able to latch onto that nucleus with a tight seal of the soma, and we can do some nucleated patch experiments. And so that is, we're able to apply a series of voltage steps, and then we can look at the perisomatic current, and then more importantly, we can isolate the potassium current for the channels are wrapped around that nucleus. Okay, and then what we can see is that when we do these experiments in the pediatric or infant range, the amount of current is right around 2 picoamps per micron squared. And then with the adult, we can actually see a much larger amount of current, averaging maybe 2.5, 3, but much more as a larger data set compared to the infant. And then we can plot that amount of current based upon age, and we can see that it starts off low and gradually increases in the adult data set. So this both of these experiments together provide us a molecular and functional correspondence to try to understand what are some of these mechanistic underpinnings of why we see this shift in the action potential. And then what I want to show here, part of patch-seq is we're able to obtain the morphology from each of these neurons. And here I have a few examples from the layer 2-3 IT3 from 3 type. The first 3 on the left are fills and 3D reconstructions from the infant, and the 2 are from the adult. And hopefully what you can see right away is that the apical and basal dendrites from these neurons are much more robust, they're more extensive, and then that is quantified here on these bar plots to the right of the basal dendrite total length and apical dendrite total length. And for the infant range, they are much higher in both of those compared to the adult range. And turned off. I'm not connected to my computer. I think this is through IT. There we go. Okay, and so again, I'm showing a volcano plot, and instead of the sodium or potassium channel genes, we identified a set of morphologically related genes. And what you can see that there's a whole host of genes that are upregulated in the adults compared to the infant. But what I'll highlight here is this PTN gene. And so this PTN gene is a cytokine that promotes neural growth and cell survival. So it kind of makes sense that in the adult that would be more diminished, and that's exactly what we show here if we look at the actual value of that PTN gene, that it starts off quite high and then drops down into adulthood. Okay, so this is kind of what I want to talk, next two slides are what I want to talk about moving forward and what we're able to do. Ed talked a little bit about the double bouquet cells, and so this is from the paper that Rachel and I put together where we're characterizing the functional properties of those neurons, and we've actually been able to find quite a few in infant, so that's one thing, one piece of analysis that we want to do moving forward is try to understand are there any functional age-dependent relationships with those double bouquet cells. We can do the same with chandelier. We don't have as many just yet, but for the few examples that we have, you can see again right away that this infant definitely looks more robust, but that's something that we want to continue to evaluate as we collect more data. One thing that I'm really excited about is looking at Cajal-Riccius cells. So these are thought to be developmentally regulated, especially in layer 1. And with these pediatric samples, we've actually been able to find a few and patch on and characterize their identity. We still need to try to do quite a few more, but we're on the lookout for each one of these cases. Since they are thought to be developmentally regulated, and if you follow the mouse trajectory, we actually really shouldn't see them later in life. But we have been able to find them in a 9-year-old, a 6-month-old, and a year-and-a-half-old. So this is still early data, but one of the things that I really want to focus our efforts on is trying to find some of these cells because they are unique. They have really unique morphologies. You can see that this one was patched, has almost 4 centimeters worth of dendrite that extended to the length of the slice. So I think they're really cool cells, and we'd really love to dive deeper into it. So this is my last slide, and I want to kind of, again, like I said, go into some of the things that we're looking forward to, excited about. And one of the things that we've been able to do with these pediatric cases is obtain resections from the hippocampus. So here's 2 examples where we have patch onto granule cells in the dentate gyrus, and then subiculum pyramidal neurons. And so these come from, we've had 2 cases so far. This top one is from a 2-year-old, and then this bottom one is from an 18-year-old. We just had 2 cases 2 weeks ago from a 6 and 8-year-old. And so for a total of these, we have 109 experiments. So still early on, but this is something that we're really excited about. Obviously, hippocampus is hard to obtain just due to the inherent nature of where it's coming from. But hopefully with 4 cases and maybe a few more, we can start to build up an idea of what's going on either with subiculum or granule cells. And then I'll just move over this real quick. This is kind of our thoughts going forward and how we are going to kind of take our PatchSeq pipeline and use this to try to really identify what are some of the functional properties and traits of each of these specimens as they come into the Allen Institute. So I talked about PatchSeq already. Histology is super important for this because all this comes from patients that have some type of epilepsy and some other comorbidities. So we really want to get a close look and try to identify to what extent the disease is pathological or normal. So that's one thing that we are working on with these histological stains, but then also that we have close collaborations with our neurosurgeon where we have frequent communication to try to identify the tissue that we're receiving versus the tissue that is resected from the pathology. This is something really early stages. We're looking at multi-patch synaptic connectivity. I think it was just brought up in the previous talk. We've done this a few times with a few of the cases. This is one of our pediatric patients, but we really want to see what's going on with the connectivity and dynamics as a patient progresses from infancy through adulthood. And then, let's see, this last one is really early stages. We've only done 1 or 2 experiments, but we want to take the tissue that we get and process them for single nucleus RNA-seq and some type of spatial transcriptomics. I say this is early stages because we are waiting for some of these technologies to emerge. Ed alluded to this a little bit about the spatial technology and the tiles becoming much bigger which would be able to support some of the tissues that we receive. One thing that I want to say about this is, if we're able to take all these together, this really gives us a unified and comprehensive view for a particular patient at a particular age, and then if there's any type of disease state that we want to try to identify. And then lastly, what I would say that out of all the 49 cases that we've had thus far, we have frozen slices of tissue from each of these patients. So once we decide on what type of technology we're going to use, we have a plethora of cases that we can start pushing through the pipeline and really start to get a good idea of what's going on. Let's see. With that, here's all my, maybe I'll just. Oh, anyways, you saw it quickly. Thank you. Yeah, no, that's a great question, and something that we would love to look at. We know that there's a huge developmental trajectory within the spine formation, spine numbers, and even the type of spine. We are able to look at that with the BioSight fills. Rachel and her team and Stacy's team is looking at an automated way to do that. Just with the number of cells that we have patched, it was just a manual approach to that would be very laborious. So we want to wait on that to see if we can have an automated or machine learning way to do that.
Hey, sorry. I have a bit of a mean question because you're obviously already looking at so many cell types, but now that you're looking at the pediatric development and you want to investigate synapses, synaptic formations, are you going to look at non-neuronal cells as well and the extracellular matrix that's kind of involved in all of this? And is this going to be part of maybe the transcriptomic part or something like that?
Yeah, so when we start doing the single nucleus RNA-seq or the spatial, we will take a look into non-neuronal cells. Specifically for patch-seq, no. But yeah, when we have the larger 10x or whatever it is, we will be able to look at that. Because I know that is a huge interest for a lot of people, especially across these developmental time points or disease-related. Yeah.
So I'm curious if you, in the 49 different patients, do you look at the germline mutations in those patients? And also somatic mutations and how that may influence the neurophysiology?
No, not specifically, not yet. But that's something that we should and could look at.
Fantastic, Doug, thank you. I was wondering from your gene expression studies whether you have some insight as to what sodium channels are involved in the young cases. I'm guessing it's not SCN8A neonatal isoform, because your SCN8 comes up as an adult. So do you know which sodium channels might be expressed early?
No, I don't. I mean, we could certainly look at that. I didn't plot or I can't remember what the trajectory looked like for that specific subunit. The sodium channels kind of weren't super interesting for us because when you look at the width of the action potential, you can quantify the upstroke rate and then the downstroke rate. And we didn't see much changes in the upstroke rate. I know that Gabor's lab in his paper that Tyus brought up, they did see minor changes in that upstroke, but didn't really invest too much of my time. It was mainly focused more on the potassium channels.
Hi, thank you for your talk. Actually, this is a question for both you and Mattis. Obviously, this tissue is probably taken from epileptic patients, right? So I was wondering whether any of you had ever looked at the confounding factor of seizure history of the patients as well as medication.
Yeah, I think that's a really good question, and that comes up with even our adult studies, because we are able to get a limited view of their clinical history, and we just haven't had the time to kind of dive into that type of metadata. And I think that's really important, for what we do at the Allen Institute as a whole is putting that data out there for the public and other researchers to use and obtain that clinical data to kind of help us guide through it, because there are so many different metadata variables that we can look at. We just, it's just with the amount of time that we have, we just can't look at all those. But we really kind of want to look to the outside resources to kind of help parse through some of those.